So it's an important protein for, it's an important protein in cancer pathway. Okay, so if you inhibit this cancer pathway protein, then there is a chance of bringing down cancer. Okay, at least some few steps we can prevent it. So it's a very notable target in cancer research. This MAP3K is a notable target in cancer research. So that's the reason we have chosen for this particular seminar workshop, right? So we have found the inhibitors of this and provided the CSV list. What is CSV again? It's a comma separated value list. So I have asked the system, I have asked the program to read the file. So we are reading through a module called pandas. Have you heard of pandas? What is pandas? Pandas is a machine learning library. Pandas is a data mining library. Clear? Okay, it's written using Python programming language. So using the pandas, I have called the file. Then after that, what I did? I asked the file to read the file, read the component IDs. So in the Excel sheet, there is a thing called component ID, right? I'm asking them to read the component ID. Okay, and provide me the SDF files. That's my request for it. I want in structured data format. SDF is none other than structured data format. Can you see over here? I'm asking in a 3D conformer of SDF. So that's my task. Can we all perform together? Once you are done with that, your next step should be click this and again press run. When you can see a star, it's in process. When the star is done, it's completed. Now I'll show you a small magic. So in this folder, how many files were there? Dear friends, please answer. In this folder, how many files were there? 7 items, right? I have 7 items. Once I press this, you can see I'm pressing run here. Once I press this, you can see that number will increase. I'm not doing anything. Can you all see? The number is getting increased. What happens there? Anyone from the group can say what is happening there?
The SDF files are downloaded for particular CID.
Correct. For each and every component ID, we are asking the software to find it and download it. So this is called automation. We no need to go and download each by clicking. This automated script will help us to download as it is. Clear now? Okay, those who have completed this download can raise your hands now. Others can lower. Only those who have completed this download can raise your hands. Good. You have to wait till the star get completed. The star refers to it's in progress. Great, that 6 people have completed. What about others? You want any clarity? Good. If you need any help, I'm ready to help. So in the meantime, you also can launch the paddle. Let it get download. In the meantime, go to paddle folder and click at this icon. There is an executable jar file will be there. Click that icon. And you can see a icon like this, paddle. Yeah, and you can see it got loaded over there. You should see like this. What I have to do is, I'm waiting for the start to complete. Yeah, it got completed for me. Once it got completed, what I will do, I'll put all these files in a single folder. I have to put only the downloads in the downloaded SDF files in a single folder. Come on, do that. I can put a heading called SDF and placing all my files inside. So nearly 49 files were there. So this is another software called PyMol. That is not that mandatory, but to visualize the molecules, I can open in PyMol. So in the beginning of the lecture, I told you about 3D structures, right? This is one of the 3D structures. Can you all see here? Yes. Once you are done with that, the next step should be opening your paddle. Come on, everyone, open your paddle. May I proceed? You are ready? May I proceed further? So it's a tricky part. Listen carefully. You can't see these 3 icons. So you have to click in the middle. Can you see 2 folders like molecule directory file and descriptor output file? Can you guys follow my arrow mark? In that, click over the center. You can see some 3 dots. So the 3 dots is a link for you to upload your directory. What directory you are going to upload now? That SDF folder. Okay, just we have placed that SDF folder in a single folder, right? All the 49 files in a single folder, right? So we are going to place that. So go to your desktop, go to the workshop folder, and that workshop folder, I have a folder called SDF, and I've selected. It might be very small, sorry for that, but I hope you can see, I guess. So the persons who have set this SDF, please raise your hands. Five people have uploaded, right? Is it so tough? Not at all. You just have to follow me. Clear? Then followed by that, give the output file name also. What name you are going to give for the output? Okay. I'm giving a name called map3k underscore descriptors. Underscore descriptors. See, there are nearly several descriptors were there, like 700 descriptors or 800 descriptors are there. Can you see all this descriptors list? Okay, descriptors I have mentioned in the beginning of the class, what is called descriptor. So that descriptor list I'm giving, like for example, carbon types or hydrogen bond count, topological surface area, all these descriptors I have enabled. Very simple, I have enabled that. Then once you are done with that, you have to click start. Yes, you can see here. Yes, you can see the process in the bottom of the slides.
Hello, sir.
Yeah,
So how to select SDF file? Can you show once from the workshop that folder?
sure, doctor. I'll show. Is my screen visible?
Yes, yes.
So in paddle, you will be having three dots. Can you see the three dots like molecule directory file?
Yes.
In that, if you click in the center, you will see these three dots.
Yes, yes,
So
I can
from these
see.
three dots, yeah, from these three dots, you can select.
We have to select SDF file, sir, from there.
Yes, sir. STF files you have to keep in a separate folder. That mandatory step you guys have to do. See, already you have made the SDF folder, right?
Yes, yes.
That folder should consist of only SDF files, not other files. So ensure that.
So we have to select it manually, sir, all SDF files.
No, sir, just upload the entire folder. Actually, this is for the entire directory. See, for example, I have just selected the entire folder and gave select. No need to go inside. You can select the entire folder itself.
Okay, sir.
Tasks, correct? So I have already provided this file to make you simple. You can drag that in CSV format. You can open that file in CSV format. You want my help? So what we are trying to do now is to align the files at the last. So we are adding the IC50 values at the last. Please open that in Excel sheet and see. See, my dear friends, I'll repeat again. There is a thing called map3 descriptors. Might be the name you have shared. Open that in Excel sheet. Open that in Excel sheet. You can see a lot of numbers over there, correct? What these lot of numbers are implying you? Anyone from the group? Is anyone from the group? What are all these numbers implying? You see full of numbers, right? What are these numbers implying?
Residues?
No, it's not residues. So I told you about descriptors. Am I right? So in the beginning of the lecture also, I have told you about the descriptors, correct? We have uploaded only the compounds. We have uploaded what? Only the compounds. What kind of compounds we have uploaded? The compounds with structures, correct? We have uploaded the compounds with structures. See, these are the 49 compounds we have uploaded in the paddle. Am I right? These structure data have been converted into what data now? Using parallel. Now recall our theory session. What we discussed. Please correlate the theory with the practical. What we discussed in the morning. What is kind of what is the mother of science?
Max.
Max. How Max help here? How mathematics help here? Mathematics helped to convert the structured data into numerical data. Clear? You all remember this? Look at here.
Yes, sir. Yes, sir.
This is what we achieve now. Let me show this. Yeah, this is the actual slide I have to show. We have converted the structures into what? Numbers. Hope you all agree with me. So what's the benefit of it?
Yes, sir.
Yeah, this is the actual slide. We have converted this structure into numbers. Now what's the benefit of it? We can give more data for my model to understand. Okay. So to view it clearly, I'll say you the options. Click at first column, go to data. Okay. And there is a thing called text to columns. Text to columns. Okay. Click that. And you can separate by comma. You can separate by what? Comma. That's why the entire file is called CSV, comma separated values. Comma separated values. Clear? It may look tough, but it is not tough at all. I'm just opening a file in my Excel. That is my Excel thing, MS Excel. Then I'm giving tab and giving finish. Now you can see it is well organized now. You all can see it is well organized now. Any doubts in it? Participants, any doubt? Please feel free to ask. Then we'll move to the next step. This step might be looking serious, but don't worry. The next step will be full of graphics, so we can easily click and play. It's kind of plug and play. So, dear friends, what doubts you have so far? Full of doubts? No clarity? What can I assume?
Dr. Chandrasekhar, I might have missed this, but the initial Excel file
Yeah.
you shared, the map3k file, you had a
Yeah.
list of map3k and the respective, corresponding chemical IDs as well as the PIC50, right?
Correct.
Was that just collated manually, or was the steps to get to that
Doctor,
point shared?
no, doctor, we have a strategy for that, but it is not disclosed outside because it's our work plan. For workshop purpose, just I presented a few of it. For
But
my
if we
individual.
were to
No, actually that cannot be disclosed now because it's a research method. Okay. We are trying to patent that method, how to find the IC50 values. If you want to be our collaborator, we want to jointly work for a research project, then we will reveal after mutual concern. As of now, we are not revealing that part, but you can manually curate. Like you can find the IC50 values for each and every molecule from PubChem.
Okay.
And a single stretch, I have given 49 molecules. I also can give lakhs of molecules in a single stretch because we have written a code for that, Python code for that.
Okay.
Once we publish the paper or we make a patent, we'll put that code in GitHub.
Okay.
As of now, we have shared only the basic codes. You can make use of that codes. Okay, doctor?
Okay, Jack.
Thank you so much. Yeah. So any doubts in the steps what we have carried so far? Can we proceed further? Is that fine for all of you? Okay. Now what I'll do, I'll share your code. I've shared a code in your WhatsApp. Please find the code. Is my screen shared? So we have made a Python code for evaluating this descriptor. Okay, that Python code I have shared. Please find it. You guys can make use of this code. We are happy to share this code with you. Only thing you have to do is you have to rename the files in your future. Look at my screen. Yeah, participants, please look at my screen. So put that code in the actual folder where you have that map3k. Okay, where you have that map3k filter CSV. Please put over there. See, there are a lot of complications over here, but to avoid complications, let me demonstrate with the initial file what I have shared with you. Later on, you can modify the columns. Can you all see this? You guys can see along with the matrix, or you can see only the code. See, in future, if you are generating a similar model, we have made a logistic regression model now. Okay, we made a logistic regression model. It's a kind of a machine learning model. Okay, and we ran the training set and test set. I told you about training set and test set, right? That we have learned it. Clear? Once we are done with this, what I have to do? Press this run button. And our prediction model is 0.84%. The prediction model is what? 0.84%. So dear participants, kindly make use of it. Once you have achieved that confusion matrix, you can share with me. Meanwhile, what is confusion matrix? Anyone aware of it? What is called confusion matrix? Yes. Have you heard of true positive, true negative? Sensitivity, specificity? Can anyone from the group can elaborate it? Yeah.
So confusion matrix is used for checking how many true positives and true negatives have been predicted by the model.
Very good. So how many true positive and true negative? Can you please elaborate what is true positive to our audience?
True positive, like we are predicting, we are giving our actual data to the model and we are predicting it, and what we have, like we have given uh, positives and negatives to the model, and the model is giving us after prediction some more positives and negatives. Then we are checking
Okay.
afterwards what all the model have predicted true. We are checking them if they are actually true or not.
Oh, great. Is it Julian you're answering?
Yes, yes, sir.
Thank you, Julian. You made it very clear. To add up Julian's point, for example, we are having, I am a doctor. I have to screen the patients who are suffering from viral fever. Okay. If I'm predicting 10 patients were sitting, if I predicted 8 out of 10, okay, that is called 8 true positives. You're getting my point? I'm predicting positive people as positive. That is called true positives. What if I have predicted as common fever as a viral fever? Then that is called false positive. You getting my point? That is called as what? False positive, because I wrongly predicted the people. What if I missed some people in between? Instead of predicting them, I missed it. What are they called? They are called true negative. Am I making sense? Is it clear for you guys? Yes. So that kind of confusion matrix you can observe in the results. Clear? Yes. Now it's time for us to move for computer-aided drug discovery. The meanwhile, what I will suggest you is install Pyrex and keep it ready. Install Pyrex and keep it ready. Everyone install. Please open the PyMol. If you are PyMol also, it's well and good. But unfortunately, if you are not having PyMol, no issues. Later on, you can install PyMol and observe our interactions later. And I also want you guys to install Discovery Studio and keep it ready. Since we are running out of time, okay, I want you to install everything and keep it ready. I know 4-hour continuous is a hectic one, but no other go. We are for a great cost here. Don't worry, my dear participants. I'll provide you a well-defined manual so that you can easily follow with me. So the actual plan of this thing is to do the docking with the values what we got. But for uniformity, what I'll do, I'll share a common protein and a ligand for you to practice. A common protein and